I'm doing well. I'm pretty busy.
Yeah.
I'm writing another paper on antibody tests. So I have
Cool.
some,
I was gonna ask you about that.
yeah, so I'm trying to bring myself up to speed on that. So I have some interesting information that I just gleaned. Somebody put together a list of antibody test papers that discuss antibody tests that have done kind of like panels of, like 1,000 blood donors, and let's all test them for antibodies and see how many were previously infected.
Hmm.
But there's a lot of problems with the data, um, even though they're not being very skeptical about it. They're just assuming the tests are correct. But
Mm.
and that sort of comes through. So
Okay.
that's important. And in one case, they found a whole bunch of old blood samples positive,
Oh,
which is impossible, right?
yeah. Right.
So we're back to the same thing with the RT-PCR test
Yep.
where you get results and it's like, well, that couldn't have happened, but it just did. So
Yep.
how do we explain that?
Cool. Well, I just fired up my recording here, so maybe we could start by would you want to give a rundown of just kind of how you got into all this stuff and what your path towards getting into uh digging into viruses and germ theory and all that, how that came about?
Yeah, so in the late 1970s, I graduated with a degree in biology and mathematics from Lakehead University. And but the mathematics part of it kind of took over, and I went on to a career in computer science and then later telecommunications from a technical perspective. But I always maintained my interest in biology. Um, and by accident in the early 1990s, I think it must have been around maybe even in the 1980s, towards the beginning of the AIDS epidemic, I heard about these scientists who didn't believe HIV caused AIDS. And I listened to this kind of intellectual program, and for the only time in my life, I wrote to the radio station and asked for a transcript, and it included some references, which I followed up, some books and some articles, and I kind of got interested in that. And for several years, I saw HIV as an exception in the world of infectious diseases. A disease that would be blamed on an infection was probably caused in gay men by the overuse of inhalant drugs called poppers, for example. And there are still scientists who hold that theory that that was the cause of the problem. But as time went on, and I met more people who were interested in this, they would occasionally tell me that, oh, you should look into mad cow disease. That's not infectious either, or you should look into polio. And I was very reluctant to do this. But when I did finally, look at some data, I realized that the environmental theory of mad cow disease, that it was caused by a pesticide called Fosmet that the British government mandated was used on cows, or polio, which was similarly probably caused by the overuse of pesticides on fruit, West Nile virus, which was probably caused by severe air pollution late in the summer, that gradually I saw that, yes, there are legitimate environmental theories. Maybe I don't necessarily reject the infectious theory or didn't immediately, but as time went on, I realized that the environmental theory had more to it. And one that is very important for today is that after 2003, I studied SARS. And the information that I gathered showed very clearly that SARS was not anywhere near as infectious as people had said, and that the response to the epidemic, the use of high-dose corticosteroids, the use of an antiviral drug called ribavirin, and the use of intubation were causing the death of many people. And it was no longer clear to me that the SARS virus played any role in this epidemic at all, that it might have just been an epidemic of doctors who panicked threw the most potent treatments at their patients, who were mostly elderly, and killed them. And so when coronavirus came along, which is SARS is also a coronavirus, believed to be a coronavirus, I realized that this was just the world repeating itself, but this time the shutdown was not just like 4 or 5 countries, but it was going to be the whole globe. But the same things were true. There's evidence that the coronavirus is not as infectious as people say it is. They're using corticosteroids again. They're using antiviral drugs again. Of course, now they've thrown into the mix anti-malarial drugs and, I think rheumatoid arthritis drugs for some reason. So they've widened the scope of the drugs, but it's the same basic philosophy. And they've been intubating a lot of patients. Recently, there was an article in the Journal of the American Medical Association that showed that in New York City, patients over 65 years old who were intubated had a 97% death rate.
Mhm.
And if that's not shocking, I don't know what it is, but there are still people who will defend that, and they'll say, well, 100% of them would die if we hadn't intubated them, so we saved 3%. But this is, I think, one of the fundamental scientific problems of today, and I guess it's a philosophical problem, and it's called the invention of data to maintain your theory.
Mm.
So, for example, this person who says that 100% would have died without intubation is inventing data because that person has no idea what would have happened. I might say, well, I think 80% would have survived without intubation. Well, I don't have any data on that because you can't rerun this experiment. Those people are dead.
Mhm.
But we need to be very careful to avoid the invention of data to maintain our theory. And this is, I see, is a very, very common phenomenon in medicine. Another example is we were talking before the show about antibody tests. So if John Ioannidis, who's very critical of the lockdown, a Stanford scientist, does an antibody test in California and finds 5% of people have antibodies, he assumes that those 5% of people were infected, had no symptoms or very minor symptoms, and were cured. But he actually has no evidence of that. He has no evidence that they were ever positive on the RT-PCR test. So this eminent scientist, who I agree with on many issues, is actually inventing data without even realizing it, because he believes that the antibody test is accurate, therefore this must be true. But he actually hasn't proven it. And if you make enough assumptions based on no data, then some of them are going to be wrong. You might be right most of the time, but even if 20% of the time your assumptions are wrong, in the end you end up with a completely unreliable theory.
So maybe we can backtrack a little bit, and you could give us some backstory on, because I know you're not the only person, talking about this stuff. Where do these ideas come from that these things might not be caused by viruses, and what's the evidence that they might or might not be caused by viruses, AIDS, all of these different things?
Right. Well, it all goes back to Koch's postulates, although I'm kind of getting tired of Koch's postulates, not because I don't think they're important, but because they were written by a bacteriologist. But basically, what he said is that if you want to prove that a bacteria causes a disease, you need to purify the bacteria in pure culture. And then you need to expose. So, for example, a pure culture you might use some nutrients in a test tube that will allow the bacteria to stay alive, right? And to multiply. And so you put a few bacteria into the test tube, nothing else. Within a few weeks, you've got a test tube full of bacteria, and then you expose an animal to it, and you produce the symptoms of the disease. And then you re-purify the bacteria. So let's say it's a bacteria that lives in your blood. So you then take blood from this animal, you purify the bacteria and make sure that you've got large quantities of the same bacteria. The problem with viruses is that virologists use viral culture, but there's no such thing as pure virus in a viral culture, because the amount of virus in the viral culture is much smaller than the number of human or other types of cells. So if you just take the cell culture material, the vast majority of what you're going to get is cellular. It is not viral. And so I want to go back to first principles and say, okay, if if a virus is causing this disease, the virus must exist. I think that's simple logic. If the virus exists, there must exist protein packages with the RNA inside them. This is a fundamental theory, and I don't think any virologist would disagree with that. That there's a protein wrapper, the famous, corona virus wrapper, which looks a little bit like the sun with knobs sticking out, and then inside that is the RNA. That is the theory. Okay, so then the third step is that you need to purify these particles. Since they exist, you need to purify them, and only then can you tell what the RNA is. Because if you put this material under an electron microscope, and what you see under the electron microscope is hundreds or thousands of identical particles, you know that your sample is pure. Then, if you find RNA in that sample, it must come from the virus. And that's the only way to know it. So what the scientists did in the start of the coronavirus panic was they found RNA that looked like RNA that had previously been called coronavirus RNA, but it was a little bit different. So parts were the same, parts were different. So they said, okay, well then it's viral because, previously people have called this coronavirus, but nobody's ever purified a coronavirus, so nobody actually knows that any of the coronaviruses are real viruses. So the common cold virus, the SARS virus, the MERS virus, there's no proof that virus particles actually exist. So they skipped by step number one, because step number one is extraordinarily difficult. And if there is no virus, it's impossible, which is my hypothesis. There is no coronavirus. Now, where does the RNA come from if it's not viral? Well, RNA is found in every cell of our body. It's found in bacteria, it's found in fungi, it's found in plants. So the presence of RNA does not prove that you have a virus. It could have come from the human body. And let's say that this RNA is created by your body when you're exposed to a respiratory challenge. So somebody, you live in a polluted area, or you have some kind of lung condition or something like that. Your body may produce some RNA that's very specific, because that's one of the differences between DNA and RNA is that DNA is with you for life. It's like a library, and the library tells you how you can make every protein in your body and how to do a lot of other things. And when the body wants to do something, it goes into the library, the nucleus of the cell, and it says, okay, I want a protein. And so the DNA is transcribed into RNA. The RNA floats out of the nucleus, and it eventually results in the creation of a protein, for example. There's many other things that RNA can do. So the presence of RNA in no way proves that you have a virus. And I'm not the only person to say this. Andrew Kaufman, who's an MD and PhD, has also said the same thing. Stefan Lanka, a virologist from Germany, who no longer believes in human viruses, has said something very similar, and I understand that he's spoken out. There's a pending publication paper from some public health scientists in Georgia that states that one of the problems with the coronavirus test is that the virus has not been purified, and therefore the test has not been validated.
Okay. So the RNA coming from inside the body, is that an accepted thing beyond viruses? Are there people that believe there are infectious viruses, but there's also is the idea of like an RNA coming out of the cell besides just like the normal? So
I don't think there's any normal RNA. Your body is prepared to react to a lot of things. Like if you go and run a marathon, is that abnormal? Well, your body is going to have to produce muscle protein and probably a lot of other things.
Sure.
So one would expect that your body would be very busy after a marathon in a very different way than, say, somebody who's struggling with heart disease.
Mm-hmm.
They may be producing RNA. The body's desperately trying to handle a heart that's not working properly. So whether you're sick or healthy, you're going to be producing RNA. I mean, healthy people eat a big meal. And of course, your body has to produce many different substances after a big meal to digest. So your body is always in a state of flux, right? There is no normal for your body, right?
Mm-hmm.
On a daily basis, you need to sleep, and that's a different state than when you get up and you eat. And then, of course, if there are disease states, then you're going into different states. Or if you exercise, or if you're in very cold air or you're in a very hot climate, then your body is going to do its best to acclimatize, and that's going to result in many changes, including the production of specific RNA.
So we have always got all sorts of RNA cruising around in our bodies at all times, basically, more or
Yeah,
less.
it's sort of the critical link between the nucleus of the cell and getting stuff done.
So how did the mainstream virologist tell between what's, uh, you know, endogenous RNA and viral RNA? I mean, I think
Well,
that's kind.
there are certain patterns of the RNA. So the RNA, like DNA, is made up of 4 different beads on a chain. And for DNA, it's ACGT, and for RNA, it's ACGU. And so there's an almost infinite number of combinations. And they say if we see this pattern, that's the start of a coronavirus. And then within that, there'll be differences because there's different coronaviruses. So that's what they say, but it's a theory that's never been proven. And the only way to prove it is to purify any coronavirus and prove that RNA that you think is coronavirus is inside those particles.
Yeah. Yeah, I guess when you break it down like that and think about the fact that RNA is doing, and I'm by no means a virologist and I don't even understand half of this stuff, but I sort of am struggling to even think of how, if we have all this RNA in our body already, how one would determine like where it started from, that they would determine, oh, this must be RNA from outside the body that's being transferred to the body as some viral
Yeah.
material and making a person sick. Like, is there a good explanation for how that was like supposed to be?
Well, virology existed before the 1930s, and the 1930s was the decade in which the electron microscope was invented. And that was the only microscope powerful enough to see things as small as viruses. So prior to the 1930s, you had an entire field of study of things that were pretty much unstudyable because you couldn't see them. So you had no hope of purifying them, because even if you purified them, you could put it under a light microscope and you couldn't tell if it was purified. You wouldn't see anything. So the 1930s should have been the start of the golden age of virology. But there's a famous paper by a famous virologist named Thomas Rivers. I think it's actually an after-dinner speech in 1936, where he says it is obvious that Koch's postulates have not been satisfied for viral diseases. So they were unable, despite this new tool of the electron microscope, they were unable to purify the virus. So the virologists could have done a couple things. One is to say, okay, well, it wasn't unreasonable to study these invisible things, but now we realize that maybe they don't exist, so we should all go get a different career. Or the second thing is you water down Koch's postulates until you can satisfy them. And that's what they chose to do. And what Rivers actually did was, what he actually said was actually pretty incredible. I'm quoting here. It is obvious that Koch's postulates have not been satisfied in viral diseases. Moreover, it is equally evident that proof of the etiological significance of viruses, that is, the ability of a virus to cause a disease, has been obtained without their satisfaction. So he's saying, who cares about Koch's postulates? We have other ways to do it. Such a statement, however, does not imply that certain conditions do not have to be met before the specific relation of a virus to a disease is established. So, okay, that's reasonable. You can't just claim that a virus causes a disease without some kind of evidence. So what is that evidence? So he says the conditions are two. A specific virus must be found associated with the disease with a degree of regularity. Now, what does that mean? What's a degree of regularity? Is that like 80%? 80% of the people with COVID disease have the virus, so therefore COVID causes the disease. Or what about the other 20%, right?
Mm-hmm.
And who determines what a degree of regularity is? Well, in 1936, that would have been somebody like Thomas Rivers. Thomas Rivers says to the grad student, well, I think you found the virus with a degree of regularity, so we've satisfied postulate number one. So there's only two postulates, and as a philosopher, you should love this one. B, the virus must be shown to occur in the sick individual, not as an incidental or accidental finding, but as the cause of the disease under investigation. So we have a proof of causation in which knowledge that something is causative is part of the proof. If that's not a tautology, I don't know what is.
So how, and I want to get into the more coronavirus stuff as we get into this more, but I just want to kind of, hammer down on this stuff before we get into that for people who might be extremely skeptical. And I mean, I don't know enough yet. I'm still just like hearing you talk about this was some of the first stuff I had ever heard about it. And then Dr. Kaufman, and the more I think about it, it's really intriguing to me, and it makes a lot of sense, like, and kind of resonates with other worldviews I have. I think one of the hard parts to kind of wrap your head around is like how this could continue to go on for so long. And obviously there's very intelligent people that have, been working on different things with viruses and virology for so long. How have we built up so much upon these, what seem to be kind of some basic assumptions in virology, and how can we explain certain things like you talked about AIDS? I'm going to throw a couple things at you so you can just kind of do what you will with this, but like you talk about AIDS with the drug use, but then we hear about all this AIDS in Africa, or we have chicken pox and people have chicken pox parties where they spread chicken pox to each other, or it's just like been so fundamental in probably the average person's understanding that there are definitely viruses and they're definitely infectious.
Mm-hmm.
We just take it for granted when there's this whole field of science, like assuming that and holding that belief and studying all these things. Like, how could all that be going on? Well, there's, like if this is true, that there is a big question mark as to whether or not, and exogenous viruses are a real thing.
Well, people assume that if people get sick as a group, that it has to be infectious. But we share an environment. And so, for example, in the winter in North America, we spend a lot of time in facilities with heated air and with less than, in many cases, less than desirable exchange of fresh air with the outside. So we're breathing in air all winter as a group that is less than optimal. Obviously there are people that go for a run every day, no matter how cold it is, but there's a lot of people that spend probably too much time. If you live in a family, then you probably eat the same food together. You have other habits. If there are certain toxic cleaning supplies that are used, those are also common. So the development of similar symptoms in related people at the same time is not necessarily proof of infection. And for every time where there's somebody with a bad cold and somebody else who's in contact gets a bad cold, there's many examples of people who were in contact with that person who don't get a bad cold. Mm-hmm. And of course, this can all be explained by inventing data, because you say, well, that person must have been immune from that specific cold virus. Therefore, they didn't get it because they were immune, but you have no information, no proof that they were immune. Regarding AIDS in Africa, it really is a completely different disease. Um, and I think we can dismantle some of the theories about AIDS in Africa. It's more common in women than in men, whereas in North America, AIDS was dominated by men, especially gay men. There was a group of fairly mainstream scientists who showed that it didn't seem to be sexually transmitted, that there was more, they believed there was more correlation with careless use of injections than there was with actual sexual transmission. And one of the factors is a disease that is more common in women than in men shouldn't be sexually transmitted because men are more promiscuous than women. The disease would be found almost universally in prostitutes, but AIDS, prostitutes are not a risk factor. They're not a risk group unless they're drug using. So non-drug using prostitutes seem immune from HIV. So why should women who are not prostitutes have a higher rate of HIV than men? It doesn't make sense. Why are there couples who have lived together for years with unprotected sex where one is positive and one is negative? Like there's many, many contradictions. So I mean, I can't completely dismantle the AIDS theory in Africa or explain exactly why every person who came down with symptoms of AIDS got that. But I can say that the unadulterated HIV/AIDS sexually transmitted theory doesn't work in Africa for a number of reasons. One of the best papers showed that they did a very good survey in South Africa. It was like a random survey balanced by the population of each area of the country. So they said, okay, this state, or however they divide the population, has double the number of people of this state. So we're going to randomly include double the number of people in the survey in this state because there's twice the population. So it was a very well designed survey. And they found that something like 20% of young Black women were HIV positive, something like 6% of young Black men, and something like 1 or 2% of white people. And so you have to say, okay, well, are Black people really that much more promiscuous, and are Black women more promiscuous than Black men? Or is there something that's creating antibodies that's causing false positives on the test? And of course, nobody ever thinks about this. And the people were also generally healthy. So if you have 20% of young Black women who are healthy and HIV positive, it's like, why aren't they all sick and dying, right? If this is a deadly illness, that they would never have known they're HIV positive unless some guy from Statistics South Africa or wherever came by and took some blood and found that out. So a lot of infectious theories crumble with the more scrutiny that you give them.
Yeah. You brought up the testing, and maybe this is a good time to get into testing. I know you've talked a lot about the PCR testing in regards to the COVID-19, and maybe you could start to break down what the PCR testing is. And also, I'm curious, is that the main method of testing for all viruses? Is that basically the go-to method for testing people for viral material?
That's a really good question, and the answer is no. I believe that PCR testing has become very popular since it was invented in the 1980s by Kary Mullis, because DNA is digital. Computers have 2 states, but DNA has 4 states. But otherwise, it's basically the same. So you can exactly replicate a chain of DNA if you have a sequence of bases in a computer. So it seems very digital, and therefore should be very precise and accurate. And so people get this idea that the RT-PCR test is incredibly simple and precise and accurate, but that's not actually the case. I interviewed a man named Stephen Buston, who's a world expert in RT-PCR, and I don't want to go into too much details, but he basically said there's 3 phases and they all have problems. So the first phase is you have to extract the RNA from the sample, because that's what you're looking for. And you have to try to get rid of any DNA that's in the sample, of which there's going to be a lot. And you also have to try to get rid of any interfering chemicals. So at the end of phase 1, you ideally would have pure RNA and nothing else, but you can't achieve that goal. So what you have is a good amount of the RNA in the original sample, but not all of it. And you have a little bit of leftover DNA and a little bit of interfering chemicals. So those are a problem for the next 2 steps. The second step is that you convert the RNA to DNA, because the PCR test itself only works on DNA. This suffers from a very low degree of efficiency. Only about 50% of the RNA can be converted. And he said that the rate of conversion varies from scientist to scientist by about a factor of 10. So you now have a completely non-quantitative test. You don't know how much, the amount of material you have at this step does not accurately reflect what you started with. You've lost a lot of material along the way. So then the RT-PCR test is a cycle, and at each step of the cycle, it doubles the amount of DNA. And they choose an arbitrary end to the cycles, say 37, and they say, if we detect DNA before cycle 37, you're positive, and if we don't, you're negative. And I looked at 33
And
different tests.
sorry to interrupt you. The cycles, meaning like it's replicating to
It's
like
doubling,
amplify.
right. It's doubling the amount of DNA at each step.
Okay.
So I looked at 33 tests that were sort of approved by the FDA. And I say sort of approved because basically the FDA right now is just rubber stamping tests. And I don't think they're doing any oversight at all. The 33 tests had an arbitrary cutoff cycle between 30 and 45. And because of the doubling, that's about a difference in sensitivity of 30,000 fold. So if you have 45 cycles, your test is 30,000 times more sensitive than a test that cuts off at 30. They're looking for different genes. If they find some but not all of the genes, some tests are okay with that, some are not. But what does it mean to be positive on RT-PCR when only 1 out of the 2 genes they were looking for is found? Like, does your virus not have that gene? And if it doesn't have a gene that's considered to be critical to the virus, how can it be causing disease? Mm-hmm. So there's a whole bunch of built-in contradictions, but if we put the internals of the test aside, there's a study from Singapore where they did almost daily tests on 18 people. And they found that the majority of the 18 patients went from positive to negative and back to positive again within a few days. So that means they were infected, they were not infected, then they were infected again, all inside of a hospital. So how do you interpret this? Like, there's no way you can really interpret this data. It seems impossible. A group from China reported on 29 people who had similar results, going from positive to negative to positive to negative to positive, in many cases, several times. I think the most maybe amusing case, and it kind of illustrates the faith with which scientists hold these tests, was a 68-year-old man in China, not in Wuhan, but in another city. He was a little bit sick, nothing serious, like a fever, fatigue, that kind of stuff. So he went to the hospital, tested positive for the coronavirus. After a few days, he tested negative, his symptoms resolved, and they let him out and put him in quarantine. But a few days later, they tested him, and he tested positive again, but he had no symptoms. So they dragged him back to the hospital, put him on drugs, antiviral drugs, even though he had no symptoms, and kept him in the hospital for a few days, and then he tested negative 4 times. So they let him out again. 3 days later, he tested positive again, so they dragged him back to the hospital and put him on antiviral drugs again. And then after a few days, he tested negative again, and so they let him back into a 14-day quarantine. So by now, the guy's probably been away from his family for a month, and he's thinking, well, if this happened to me 3 times, maybe it's going to happen more than that. And I'm basically in prison until this is over. But the worrisome thing from a scientific point of view is that the authors of the paper never questioned the results of his test. They never said, maybe this could be a false positive. You know, he's got no symptoms, he's testing positive, maybe it's a false positive.
And the idea that it could be a false positive is not like a fringe idea. I mean, it's well known that there is a chance for false positives, correct?
Right. I looked at these 33 tests on the FDA, and probably half of them mention false positives. They say, one of them says any test can suffer from false positives. Another says false positives could be caused by the following reasons. They also say we've not tested this test on asymptomatic people. So, and a study in China estimated that if somebody who was asymptomatic tested positive, there was an 80% chance it was a false positive. So this is a big issue, right? This man who left the hospital, he was asymptomatic. So according to this paper, there was an 80% chance that his positive test was false. And yet the scientists obviously never considered that possibility. They treated it as if the test is the gospel. The gospel says he is infected, bring him back to the hospital.
Is this where the idea comes from of all this confusion about, well, maybe you can't get an immunity to it. Maybe you get reinfected by it. Maybe I don't know if you've heard that. I'm sure you have.
Yeah, I mean, they've been twisting themselves in knots. Another example of what's been going on is that in China and Korea and probably elsewhere, they've been monitoring people who've been let out by this 68-year-old guy, and they've been finding a significant percentage of people. I think in China between 5 and 15%. A significant percentage are later testing positive, usually without any symptoms. Again, so they have no easy way out, because they could say, okay, well, the test has a big problem with false positives. But if it has a big problem with false positives after you're out of the hospital, well, maybe your positive test when you went into the hospital was false. I mean, there's no reason to think it couldn't have been, right? So if they start saying, yeah, there's a lot of false positives, now they're stuck because they have no way to validate the test. They have no way to say when there's somebody who gets tested because they don't have symptoms, but maybe they were in contact with somebody who did. So they test them, they're positive, and it's like, well, is this person infected or not? We have no way to say. It could be reinfection, but if it's reinfection, it means that it's futile to do social distancing, because eventually people are going to have to be let out, and everybody's going to infect everybody else anyway, right? Even if they've already been infected 5 times before, maybe they're still going to get infected again. So they don't really have a good way out of this conundrum. I mean, I do. I can say the test, there's no virus and the test is meaningless, and my theory is completely consistent. But they're tying themselves in knots. Eventually, the Koreans said, these are false positives. But they're trying to maintain the position that a test after you've been infected is a false positive, without questioning whether the people that they're diagnosing as infected are also false positives.
So all these tests that we're referring to that aren't antibody tests, so like the current COVID-19 tests that are taking place that aren't antibody tests, are they all PCR based?
I don't believe there's any other tests. I have heard some references to rapid tests, because PCR just take a little while, so it might take a day or two to get your results. I have heard about some on-the-spot rapid tests, but I know that in my province of Alberta, they had to withdraw all the rapid tests because they weren't working. So I don't think there's at present a good alternative to the RT-PCR, and I think most people who are being tested positive, there are people who are being what they call presumptively diagnosed, and this is occurring a lot with old people in old folks' homes. They die in an old folks' home, and they're recorded as a COVID death, but they never had an RT-PCR test.
They never even had the inaccurate test.
No. There was an article in New York, a retirement home in Brooklyn, 55 people died, and towards the end of the article it said, due to all of the constraints and the difficulty and blah, 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 unfortunately, nobody in this retirement home has yet had a test, which is an admission that all 55 had not been tested. In Belgium, they're recording all retirement home deaths as COVID, even though only 5% of people in the retirement homes have been tested. I just read something from England where they've completely changed how death certificates are done. And so now, basically, whereas before a doctor would record the death certificate and then it would go to the coroner who would certify it, now the doctor can sign the death certificate, and you used to have a relative or somebody who knew the person, sort of co-sign the death certificate, so sort of as a safety check. Like if the doctor says this person died of a heart attack, maybe the relative would think that no, it was malpractice, and would refuse to sign the death certificate. So it was kind of a safety check, because in England they had a massive scandal where there was a mass murderer who was a doctor who was signing death certificates, who was putting on false causes of death, and thereby covering up all the people he'd killed.
Wow.
And so that's why they strengthened the death certificate process. And for COVID, they've completely gone back to the old way. In fact, it's now worse than it was before. For example, the funeral home director can sign the death certificate where a relative used to sign it. So, and the doctor who signs the death certificate actually may have never seen the patient. So they're told, this person developed a fever and a cough, and then they died. And okay, COVID. He signs it, the funeral director signs it, and we got a statistic, another COVID statistic.
Yeah, I've heard similar things about the CDC in the US sending out memos to hospitals telling them to just kind of assume, if there's a death that seems like it's COVID, call it COVID.
Yeah, there was they sent out a memo, the National Vital Statistics System sent out a memo with a new code that you could use to record a coronavirus death, and it said this code should be used in the case where a death is known to be caused by the coronavirus or is associated with the death of the patient. So basically, everybody who tests positive should be recorded. And then, I think it's true that it is true that a lot of people who've never been tested are just being assumed to have the coronavirus, despite the fact that there are no symptoms of the coronavirus that distinguish it from a million other illnesses.
Yeah. So one more quick bit before we move on from the PCR stuff is, I know you mentioned Kary Mullis, and he kind of invented the technology, right? But he himself didn't intend for it to be used as a testing technology. Is that right?
Well, it's a little bit complicated. I think he developed PCR as a manufacturing technique. He wanted to manufacture DNA, so he wanted to start with one set of DNA in the morning, and he used primers to kind of snip the DNA and duplicate it. And so by the end of the day, you would have 1 billion copies of the portion of the DNA that you wanted. And so that's what he was after. He was not developing a test technology. But on the other hand, the fluorescent probe that is critical to the RT-PCR test, because to test if somebody's positive, you can't count DNA directly. So what they measure is the amount of fluorescent light that's received from the sample. And this is due to these fluorescent probes, which were invented by Kary Mullis. So if Kary Mullis didn't believe in the use for testing, he developed one of the critical pieces that was needed. But I know Kary Mullis did say that quantitative PCR is an oxymoron. And this Stephen Buston that I interviewed confirmed that RT-PCR, without some additional complexity, is not quantitative. And yet the cutoff to divide positive from negative is quantitative in the sense that the cycle number is assumed to be referring to the amount of DNA. But what Buston is saying, and what I think Carey Mullis was saying, is that you can't actually say that. So if I have a cutoff of 37 and you have a cutoff of 37 in a different lab, we're going to get a different number of false negatives and positives because our numbers are not really the same. Whereas the number 37 is the same, but the quantity that it's measuring based on our different procedures, we use slightly different chemicals, we have a slightly different machine, we have slightly different procedures, we collect the samples in different ways. You know, there's like 50 differences between our two labs, and that will result in significant differences. So I may have, you know, be getting a higher percentage of positives than you, but that's not because I have more infected people. It's because my test is a little sloppier than yours. And I don't think it's as simple as saying, you know, there'll be more false positives or more false negatives. It could be that you would be measuring some people positive, that I measure negative, and vice versa.
Mm. Is there anyone in the mainstream, you know, kind of addressing this stuff or questioning the validity of the RT-PCR's use as a testing mechanism, or?
Well, Stephen Buston said that he had developed guidelines for RT-PCR in general, called the MIQE guidelines, M-I-Q-E, and he said he was talking about developing guidelines for coronavirus testing. Now, he doesn't question the viral nature of the RNA, but the RT-PCR test is only an RNA test. It doesn't have anything to do with where the RNA came from. So it's possible when he looks at the mess of testing that's out there, he might say some critical things about the quality of RT-PCR testing.
So now maybe what about the antibody testing? Is the technology for that somehow more accurate, or could it give us a better picture into, what are the antibodies actually? What are they, and what are they showing us?
Okay, well, there's two basic type of antibodies called IgM and IgG. The first thing I should say is the only way to know that you actually have antibodies to the virus is to purify the virus, to expose an animal to the pure virus, and to verify that the antibodies only appear after exposure. So if you do this on 100 animals and you test for the antibodies ahead of time and they're not there, and you expose 100 animals to the virus, and then 99 of those animals have antibodies afterwards, you say you have a pretty accurate, you've got the right antibodies, and it seems like there's good accuracy. So the theory is that the IgM antibodies are kind of generic antibodies, and they're produced very quickly after infection, within a few days. And after you're cured, after the virus is being defeated, the IgM will start to decline and will eventually disappear. IgG, on the other hand, are very specific antibodies, and they take longer to develop. So in the first few days of infection, you might not have IgG. But after the infection has been resolved, after there's no virus left, the IgG antibodies will persist. And this is the kind of lifetime immunity that we're talking about. So theoretically, if you had chickenpox, you should have IgG antibodies in your body to chickenpox, but you won't have the IgM. So they believe that by looking at the RNA and the IgM and the IgG, they can tell what stage of the infection that you're at. But I think that's actually very difficult. And if we're talking about these surveys of the general population, I mean, let's say you do a survey and 5% of the people in Sioux Falls that you test positive for IgM or IgG. So you're going to say, okay, those people were infected. They might not have realized it, but they were infected and they cured the infection, and they're now on the, if they're not immune, they're on the road to immunity. The problem with that is that you actually don't know that they were infected. You don't have RT-PCR tests. You don't have purification of the virus. You have no information. And for the 95% of people who aren't testing positive on the antibody, you don't know for sure that none of them would have tested RT-PCR positive if you tested them. Like, to actually validate the antibody test positive would require that you monitor a lot of people, and then you'd be really excited when some of those people become coronavirus RT-PCR positive, and then you start monitoring for IgM and IgG and seeing how many days that it takes. But most of these studies are in no way set up to monitor this. And I've been looking at a bunch of antibody testing studies, and I've noticed two very interesting things. In one of the studies, they were looking at blood donors, and they actually had access, for most of the donors, they actually had access to old blood, presumably from a year ago, but before the coronavirus was believed to have existed. And they found 14% of those samples testing positive. And they said, oh, well, we have a big problem with false positives because that helps to maintain their viral theory, but they don't know that they were false positives. Maybe the virus was in circulation 2 years ago. And until we actually noticed it, we didn't freak out. And it's actually our freak out that's causing all of these problems.
Yep.
Another problem that I noticed, which aligns with some a surprising amount of research on air pollution, is that in New York State, they provided the percentage of positive antibody tests in each region of the state. And in the regions of New York City, Long Island, and Rockaway, one other part of New York State that's close to the city, they had 10 to 20% of people antibody positive. But in the rural areas of New York State, they had 1 or 2% positive. And so there seems to be a very strong correlation with air pollution. And this correlation has been found with the likelihood of testing RT-PCR positive, of having RNA in your body, and also the risk of death from the coronavirus. So there seems to be a really strong correlation between all the testing and all the outcomes and air pollution. And if you think about it, Wuhan has a big problem with air pollution. Lombardy, Italy has a big problem with air pollution. And of course, New York City as well. And these correlations that these air pollution researchers have found have been quite striking and quite strong. These are not just minor correlations. And so my theory that maybe the RNA is produced in response to a challenge to your respiratory system, it fits in with this. Like, if you're exposed to air pollution, you may be more likely to produce the RNA that is mistaken for a virus. So this is, now I think what people will say about the New York data is, well, New York City was the epicenter, it's highly congested, there's more infections because there's more people in a small area. And it's not an unreasonable hypothesis, but there's really no more evidence than that, than saying that there's a correlation with air pollution. And given that there's a correlation with air pollution in the RNA test and bad outcomes, it seems like the air pollution hypothesis is not unreasonable at all.
Yeah, I mean, that makes a lot of sense to me. And when you think about the type of stuff that's in air pollution, I mean, even just calling it air pollution, it sounds kind of vague, and you just imagine, oh, well, the air isn't super clean, but you imagine all the brake dust and,
Right.
industrial chemicals. And I mean, I can't even imagine of what kind of stuff, composes this really bad air pollution.
Right. Well, air pollution researchers often break it down, right? Like they'll measure the amount of particulate matter in the air, basically dust, which can be quite toxic because, as you say, it could be, particles from a brake, or it could be other, things created from chemicals, like flakes of paint that end up as a particle of dust. And then there's certain chemicals like nitrous oxide and ozone and other things like that. There's volatile organic chemicals. And a lot of these things are measured in places that have air pollution problems. And so they've looked at some of these individual risks. And in some cases, certain risks are not correlated with COVID and other risks are, right? So there is a little bit of more specific data rather than just saying it's air pollution. So
Sure.
I'm being a bit sloppy there because there is actually some, like particulate matter definitely seems to be related to all the COVID indicators.
And so this would be kind of um, the terrain theorists' kind of view on these infectious diseases, right? Primarily, and there is some precedent for this as well. Like, how accepted is this? Because I've heard this story a couple times, and I haven't looked deeply into the details to try and validate them myself. But you mentioned earlier polio, and I thought I had heard that there was some wide usage of some sort of chemical, potentially containing arsenic. I don't know if it was DDT or what. I don't remember, but I know I had heard that that was potentially responsible for the symptoms that were thought to be polio, and that it was sort of intentionally used as a cover-up rather than kind of admitting to this pollution exposure. So I'm wondering if there's evidence, how much evidence there is for all of that.
I can quickly describe. There's really two phases of polio. There's pre-World War II and post-World War II. Pre-World War II, pesticides were generally based on arsenic, sometimes also lead. So you can imagine that pesticides in those days were extremely toxic. It's also known that polio has a very strong seasonality, and that the season of polio matches the fruit harvest season. So that means it's late summer to early fall. And this was noted in papers from the 1930s, where they had these beautiful graphs of different states of the United States, and they would superimpose the number of polio cases and the harvest of all the different fruits. From whatever cherries at the beginning to apples at the end, right? All the different fruits. And probably 90% of the polio cases occurred during this about a third of the year when fruit was being harvested. So that was pretty strong. Now, after World War II, organochlorine pesticides, especially DDT, became widely used. DDT was seen as a panacea. You could buy underwear with DDT in it. You could buy paint with DDT in it, because it was believed to be a completely harmless compound that would kill all instincts. They fogged cities with DDT. They sprayed cows with DDT. They sprayed fruit with DDT, right? Like, it killed all insects. It was wonderful, and it had no side effects until they found out it was killing people. And that it actually is quite toxic. There are still people who say DDT is not toxic. Well, that's not true. I have U.S. government documents from the 1950s describing the toxicity of DDT. But people fell out of love with DDT relatively quickly, and that sort of parallels the epidemic. So the peak of the epidemic was 1952. The vaccine trial was 1954, and when they initiated the vaccine for a couple of years, when they initiated the vaccine, the number of polio cases had dropped dramatically already. It was less than half by 1954. By the time they really rolled out the vaccine, it was about 5% of the peak. Then there was a couple of years, I think 58 and 59, where the number of cases went up. And there was a scandal known as the Cutter vaccine scandal, where they had to take the Cutter vaccine off the market because it was causing polio. But by 1960, when polio had pretty much disappeared, there was a plaintive article in Journal of the American Medical Association. Well, it must have come out after 1960, but shortly after, saying that in 1960, less than 50% of the vulnerable population had been vaccinated. And yet the polio epidemic had disappeared. So clearly the vaccine did not eliminate polio. but the rapid reduction in the use of DDT, the more careful use of the pesticide, may have been what did the trick. It's hard to see because it's hard to say because a lot of the use of pesticides like DDT is undocumented, right? You know, if a city sprayed the streets 5 times during the summer with DDT versus 1 time versus not at all, like where would you find those records?
Yeah.
It'd be very difficult to get that.
And so it's kind of funny hearing some of that type of stuff about some of the crazy things that humans have done in the past. And somehow now in the present, we think we've got something figured out that we didn't before, you know, and the idea that, you know, spraying crops with glyphosate, for example, I don't think that's tied to, you know, any necessarily viral outbreak that I know of, but I'm just saying, you know, how many things are we doing right now that could be associated to various different types of illnesses that are happening? And as soon as something gets called a name and there's a theory behind, you know, it's being caused by a virus, you know, it's easy to kind of have this, to create an idea of what's causing something without really knowing. I just everything you're saying, it makes a lot of sense, and it starts to feel like, well, it's not quite so clear what's actually going on, you know?
Well, I mean, if you look at the North American diet, which starting in the 1950s, North Americans were pushed towards a low-fat diet, which has proven to be a disaster because a low-fat diet is a high-carbohydrate diet. The high-carbohydrate diet has caused a massive increase in, you know, heart disease, things like that. I think most people now, there's still some true believers, but most people now know that that was a disaster brought to us by the medical system, imposed on us, you know, propagandized by doctors. You know, you need to eat less fat if you want to lose weight. Well, I mean, it doesn't work you can't lose weight on a high carbohydrate diet, right? I mean, it makes sense. If you have less fat, if you eat less fat, you will be less fat, but it doesn't actually work that way. But that was a massive health disaster brought to us by the same people who are bringing us the coronavirus disaster. And so the fact that the majority of medical doctors in the 50s, 60s, 70s, 80s, 90s believed in the low-fat diet doesn't make the low-fat diet right. And it doesn't mean that if there's a majority of medical scientists who believe strongly in the coronavirus theory, that doesn't make the theory right. Like like often with global warming, it's cited that 97% of climate scientists believe in global warming. That is a completely irrelevant statistic. Even if it was true, which I don't believe it is, I mean, it's more than possible that the 3% who don't are correct. You know, how many Einsteins were there when he came out with his theory? There was one.
Yeah.
Right?
Yeah, that's a great example.
Said, you know, this guy's crazy. I have an interesting computer science paper that shows that all the major advances in computer science that were put into the form of a technical paper were rejected in a brutal fashion. Like, this is completely impractical, or this is way too complicated, or something like that. Like they were scathing reviews of what turned out to be major advances. Like Alan Turing, for example, had a paper rejected that became one of the fundamental foundations of theoretical computer science. I mean, this happens in every science. People talk about, well, it's not peer-reviewed. Well, the biggest advantage of peer review is that it suppresses innovation. And it keeps the establishment clique in power, because the establishment clique does all the peer review because they're in the majority. And if somebody comes up with a radical theory, it gets crushed and it cannot be published and it cannot get grants. And hopefully that person learns their lesson and they go away and lick their wounds. They leave science and they do something else because they realize they're not going to get anywhere. And that's why I don't even try to I have occasionally had papers peer reviewed, but I normally do not try because I know that my ideas are so radical that they're too radical for scientific publications, which is an indictment of science. There should be nothing too radical for some scientific journal to say, well, this is kind of crazy, but the theory kind of hangs together, so maybe we should publish it and see what other people think. But the journals are so scared that it's not like people are going to come up with reasonable arguments. It's like people are going to yell at them and say, how irresponsible of you to publish this. This is total garbage. Like, who are you? Let's shut down your journal. Like, that's the kind of response, not like, well, this guy is wrong because we have purified the virus, right?
Yeah.
That would be a good response. But they won't do that. They will say, everybody knows that Koch's postulates were developed in the 1800s, and we don't have to satisfy them anymore. They're outdated. There will be arguments that are irrelevant, or there'll be ad hominem attacks. Where is his PhD? Things like that are used to suppress innovation.
Mm-hmm. Yeah, and for me, it can be a little bit tricky to let go, or even just to experiment with allowing yourself to temporarily let go of something that's been so ingrained in the minds of everyone, like viruses being an infectious disease. But when you look at all the other ways that we are currently wrong and have been wrong and potentially are wrong in the medical field, in the dietary field, like you mentioned, things we're doing in agriculture, like, tilling fields and using massive amounts of pesticides and herbicides and all of this stuff. And I just look around me in the kind of establishment mainstream, for lack of a better term, and there's so much that seems to be wrong and not effective and maladaptive. And so there is a large precedent, I think, for the possibility that there is a misunderstanding in something that seems to be so fundamental,
Yes. Another analogy that I have is if you went to, some average soldier in the U.S. military and said, does the U.S. military make America safer? I think you'd normally get the answer, sure, right? And you'd probably get that from 99% of the U.S. military. But anybody who's looked at American foreign policy and the results of American wars would realize that the U.S. military makes Americans less safe. It makes them targets when they're overseas. So you have an entire institution that has a belief that is necessary to be within that. I remember reading about a priest who, at some point in his life, had a major psychic crisis and decided he didn't believe in God. But he kept on as a priest because that was his job. He just kept his mouth shut. But he no longer had that fundamental belief because he understood that in a hierarchical system like a religion or the military or the current medical system, you have to maintain the core beliefs. And if you stand up and say, okay, I'm a doctor, but I no longer believe in vaccination, well, you can expect to lose your licensure pretty quickly. You're not likely to survive.
Yeah. So, just you bringing up vaccinations makes me think. So if you're right and the other people that are kind of talking about this type of stuff are right, then what does that mean for vaccinations? Because don't they completely depend on, infectious viruses being real to be effective in the first place?
No, because vaccines are not tested for their ability to prevent disease. They're tested for their ability to generate antibodies. So we, with the antibody tests, scientists all agree that compound X is a coronavirus antibody, and they come up with a vaccine that produces compound X in people who are vaccinated. And then they say, okay, you're protected from the coronavirus. There's absolutely no evidence you're protected from the coronavirus if it exists. But that's the level. I mean, in many cases, it would be impossible and very time consuming. But on the other hand, if we want good science, we need to have vaccines that are shown to actually reduce the incidence of the disease. So it's perfectly plausible that they can produce a vaccine. I mean, I read a paper the other day, it's a few years old. It said that the average time to produce a vaccine is 10 years, and the success rate is 6%. And this is within a system which is very lax in terms of adding vaccines to the schedule. So what happens if the first coronavirus vaccine doesn't take 6 months? It doesn't take a year. It takes 5 years. And then when it gets into phase 3 testing, you know, it's so obvious that it has problems with adverse effects that it just can't be marketed, right?
Mm-hmm.
We could be 5 years out from now and still not have a vaccine. Because although the vaccine doesn't have to stop you getting the coronavirus, it does have to produce the antibodies, which is not necessarily all that easy. And secondly, it can't have too bad an adverse effect profile. I mean, you can shove some adverse effects under the rug, and they do that all the time. But, you know, if you have children dropping dead within an hour of being vaccinated, you have too many of those cases, then it's going to become obvious this vaccine has a big problem. And there are concerns that they will be using an RNA vaccine because now they're so reliant on the RNA for everything. And this is pretty unproven and pretty dangerous technology. So whether they'll be able to get away with something that's sort of reasonably safe and produces antibodies is highly questionable.
Yeah. How much have you been looking into the RNA vaccine? Because I've been hearing that it is pretty experimental and that it's kind of all along the lines of genetic engineering, that it actually gets into your.
I mean, that scares me because, you know, in AIDS, the drugs, we're not talking vaccines with AIDS, but the drugs got into your cells and interfered with DNA synthesis. And that's why they were so highly toxic. And so now we're talking about putting RNA into your cells, and the RNA is going to, like, I don't know, modify your DNA, or I don't know how this works. I mean, will it work, or will the cell just reject the RNA because it knows that it's not the right kind of thing? Or will it actually get into some of your cells, and could that cause the destabilization of your genome, which could cause cancer? Like, the number of potential possibilities are great. But I have not been looking at the vaccine. I have enough things to look at with the basic disease model, the existence of the virus, the RT-PCR test, the antibody tests, the reasons why people are dying. Because that is a question I got a lot at first was, well, there must be a virus because everybody's dying.
Yep.
But when you actually stack up the reasons for people dying, it's like average age 80, 1, 2, or 3 or more pre-existing health conditions. They live in an area with air pollution. They're being given experimental drugs. They're being intubated. Like, you add up all the possible causes for death, and they're pretty significant and really severe potential causes for death.
It's not uncommon for an individual to have an experience of, you know, hypochondria, you know, thinking that they might have some sort of disease or something, and kind of overreacting and being potentially open to some type of treatment, or just going through a thought process of like a panicked episode of thinking that they have a disease. So is it really that unreasonable to imagine that like a group of people or a collective or potentially a percentage of the entire world could have that type of experience?
No, an analogy that I thought of was the Japanese Exclusion Act. I think there's a lot of parallels. This was put in, I think, by Roosevelt during World War II, and it forced all Japanese Americans to be put in concentration camps away from the coast. because they were believed to be a danger to American security. So all the politicians, except with the exception of maybe one or two radical communists or something, were in favor of this. The media loved the story. Like every fake story about some Japanese person wanting to sabotage America was great news. And very quickly, everybody in America hated the Japanese. You know, they've never seen a Japanese person because at that time they were pretty much clustered on the West Coast. So as an example of a belief that took hold throughout the entire population, there was no resistance outside of the Japanese in terms of this is an outrage, this is a violation of their human rights. There's no evidence to support this. There was hardly anybody doing this. I'm sure there were a couple of civil liberties lawyers who vainly fought against this, but the vast majority of the population of the United States just sucked it up, and Canada did the same thing. So all of North America just believed this because they were told by politicians and they were told by journalists that this was the case. Mm-hmm. And this is the same thing here. We have the politicians saying that we should go home. You have the journalists harassing the politicians, saying, you didn't send people home soon enough. The conditions are not strict enough. Everybody should wear a mask. And, you know, for a long time, journalists were pushing politicians in the one direction. Now I think it's turned around a little bit. And you've probably heard that yesterday, the leading mathematical modeler in the United Kingdom, Neil Ferguson, was deposed after a sex scandal, which involved, yes.
I didn't hear this.
Yes, no, it turned out he had a lover who was a married woman, and she was traveling across London to visit with him, breaking quarantine. And so the fact that he had encouraged his lover to break quarantine, not the sex scandal, was what brought him down. So he's been forced to resign. But there's a lot of indications that the whole construction is starting to crumble. There's a lot of resistance in the United States. There's been big demonstrations.
Yep.
There's been demonstrations in Canada, some in Europe. There's more and more resistance. There's that hair salon owner in Dallas, I think, who opened up her hair salon. And the judge said, if you apologize, then I'll let you off without jail. And she said, why should I apologize for providing work for people who have no money? My employees have not earned money. So I'm not going to apologize, and put me in jail. So they put her in jail. And of course, within 24 hours, they'd raised about $100,000 to pay for the $7,000 a day fines that she's incurring. And then in a week, the governor of Texas has said in a week, you can open up hair salons. So in a week, what she's doing is not illegal. So it's just,
that's silly.
It's just bizarre, but there's like no common sense. You have mothers or fathers playing ball with their kids in the park being arrested in an empty park. You know, you'd look at it going, well, there's nobody here. So who are they transmitting the virus to? There's absolutely no common sense. It's become totally bureaucratic. But I think more and more ordinary people are saying this is just crazy. And they're seeing that in the places where there's strict lockdowns, the results in terms of the number of deaths are worse than in places like Sweden, where there were no lockdowns.
Mm-hmm. So, I mean, do you have any, besides just sort of like fears running wild and people panicking, I mean, do you get into thinking that there's any sort of nefarious intention behind any of this? I mean, not that there really even needs to be, but I'm just wondering what your perspective is on that.
Well, I don't think it was a conspiracy, but I think the public health officials had built a beautiful pandemic machine. And they had been trained. Like there was a training exercise, I think in New York, run by Bill Gates, on what to do in a pandemic. So they were simulating what you should do. So just like an airline pilot, everybody in that, they were all leaders. I mean, these were probably not like nurses on the floor of a hospital. These were like hospital managers and public health officials, people like that. They were told exactly what to do. You need to buy personal protective gear, you need to quarantine people, you need to do all of these things. So when somebody pressed the red button that says pandemic in China, then the whole world of public health just went into pandemic mode. Politicians and journalists have learned long ago that you don't argue with medical officials. Like a journalist who publishes articles that are opposed to vaccines will lose his job, his or her job. A politician who speaks out against vaccines or drugs or some other aspect of the medical system will find that at the next election, they have a well-funded opponent who's being funded by the pharmaceutical industry, and they'll lose their job as well. So politicians and journalists are incredibly compliant. And so when the public health officials said, we're all going to jump off a cliff because it's necessary, and we want you to herd up the entire population so we all jump off together, then they just did what they're told. And they walked down this path. Well, let's just destroy the entire economy. Let's not worry about the consequences of destroying the economy, the suicides, the domestic violence, the people descending into alcoholism and drug abuse, the poisonings from careless use of cleaning materials and disinfectants. Let's not worry about any of that. We've got this one foe in our life. Let's cancel all surgeries at hospitals. Let's stop people going to the hospital to get the necessary physiotherapy to allow them to live a life without pain, or to go to an acupuncturist or something like that. Let's just all jump off the cliff because we're so scared of this coronavirus. And everybody just followed along. But there are definitely a lot of commercial interests who are taking advantage of the situation. Like if you're a vaccine manufacturer or a drug manufacturer, as soon as this happened, you must have been having meetings about, okay, how are we going to be first to market with a vaccine or a drug? I mean, if you weren't, you wouldn't really be very smart.
Yep. Yeah, that all makes sense to me. So do you have a firm belief personally on the whole, do you believe that viruses don't exist as we think of them, and they are just sort of endogenous DNA or RNA that's created in our bodies to sort of be messengers for exposure to toxicity and other things along those lines? Or
yeah, that's kind of what I believe, and I don't think they're very reliable markers. Like given the number of asymptomatic people who are RNA positive, it's clear that even if the RNA is representing a respiratory challenge, it's not necessarily that significant in most people. So I don't think that these markers that we have really are terribly useful. Like it's not like we can change from saying, okay, the RNA is not from a virus, but it tells you that you have a respiratory illness. Well, it obviously doesn't, because most people don't have any kind of illness when they're positive. But I do believe that the whole field of virology is rotten, and we need to end it. I believe it's been going on for thousands of years, that we've had this belief in invisible infectious agents. It's ingrained not just in our lives, but it's ingrained in the stories that are passed down from medieval times, from Roman times, which were probably based on stories that were handed down from like the Persian Empire, other empires. Like I don't think you can go back far enough in time to get away from this belief, because it's a way for people to explain, you have a bunch of horses in the field that get sick all at the same time. Well, somebody put a spell on them, or somebody, some mysterious substance poisoned them, right? Like if you have an invisible substance that you can just draw on whenever you want, you can explain anything. And so it may have kind of seemed like somewhat more rational than magic, right?
Yeah.
Once people got past a witch put a spell on my cows, it was like my cows got a virus.
So when people say like, we have a biome, we have all these bacteria inside us, and then they also say, and we also have a virome. We have all these viruses inside our body that are sort of essential to us, you know, various processes in our body. What are they talking about there? Are they just
Well,
talking about RNA that's,
well, we
coming
know
from our
that there are
cells? Or?
exosomes, which are RNA that's packaged in our body, and we have limited understanding of what they do. And they probably perform important functions.
Mm,
But that doesn't mean that they're exogenous. I mean, I
sure.
reserve the word virus to something that comes from outside your body and that causes, at least in some people, causes symptoms. The Latin word virus means poison. Um, so it gets very confusing if you talk about endogenous retroviruses. I don't know really what you're talking about because you're
Mm,
talking about kind of like an exosome, something that never left your body. You were born with it,
mm,
and if your body is severely challenged, then the theory is that it can cause some disease. But it's like your body's being severely challenged. So do we need anything beyond the challenge of your body with pollution or radiation
yeah.
or mental stress or something like that?
Yeah.
Do we need to invoke some kind of virus? But I mean, I think we probably should spend more time. The virologists should go on and study exosomes, and they'd probably learn a lot of interesting things about exosomes, and some of them might, help people obtain better health. The problem with modern medicine is if there's not a commercial product at the end
Yeah.
of the tunnel, then nobody's interested. So if we can have, drugs that enhance our exosome, then that's something, right? But
Yeah.
is it actually going to make us healthier? I'm skeptical.
Yeah. So do you know, can we pass exosomes? Like, can they come out of our coughs and spread from one person to another? Or
I don't believe so. But I have, that's not something I've really studied. Maybe that's possible that in a non-pathogenic way that we could pass these things on and thereby transfer maybe immunity or something like that. So
Mhm.
I'll remain neutral on that. I mean, it's an enticing possibility that our bodies, that through close physical contact, that we could actually enhance each other's immune system. It's a nice theory and one that I'd
Yeah.
like to believe, but I don't have any information.
Well, I think I've also heard somebody, theorize that maybe, it's kind of along the lines of an infection kind of model, but it just has serves a different purpose, which would be that they could spread from one person to another person to carry the message of like, hey, there's something toxic in the environment, like get your body prepared for, exposure or whatever.
Yeah, well,
I don't.
again, it's not something I know anything about,
Hmm.
but I mean, one thing I've learned, I've always been, um, I think, an over rationalist. Always trying to break things down, but at the same time, I've understood that there's a lot of things that can't be broken down. Like I'm not a reductionist in my approach to science. Like I like to break a problem down into pieces, right? Like when I'm looking at coronavirus, I'm going to look at like what are the pieces of the coronavirus theory I want to address. So in that sense, I'm a reductionist, but I understand there's a lot of things that happen um, in the human body that cannot be broken apart. So for example, when some hormones go throughout your body, they affect many parts of your body simultaneously. And so trying to isolate one thing becomes impossible.
Mm.
And I've accepted that for a long time, that there are things we will probably know because our science tries to do experiments with one variable. But with the human body, it rarely is that there's one variable, or
Mm.
you're missing something. Like you might study, okay, so you have a drug and a placebo, so okay, you've got one variable, but then you want to study that drug, and now you have every organ in the body as a possible target. So you've essentially blown up your research into a very complicated job. So what people do is they say, well, this is a heart drug, so we're going to study the heart, and maybe the liver could be a problem, so we'll study that too. And then they ignore other parts. And then later you discover that the drug causes kidney failure because nobody looked at that, right?
Mm-hmm. Yeah, the body's not just a machine where you can separate one part from the other and think that, that's going to solve a problem. It's, a complex system of interconnected, functions.
Mm-hmm. Mm-hmm. Yeah, it is. And it's like the comparison between the brain and the computer breaks down because a computer is something that you can analyze in a reductionist fashion, right? Like you can take a computer and you can change one bit and you can cause something different but predictable to happen. With the human brain, there's no equivalent of a bit, right? There's no, like, I don't think we're ever going to find that a thought or a memory is stored in sort of a little, cubic millimeter of your brain. And that if you remove that cubic millimeter, then you forget that one person or something like that. Like I don't believe it's true. And besides, how are you ever going to do an experiment on the human brain where you're actually removing little bits of the brain to see what happens when you remove 1 cubic millimeter at a time? Like, it's impossible.
Yeah. Well, I want to thank you for coming on, and I appreciate your podcast called Infectious Myth, which I would encourage people to check out. And I'll put that in the show notes and everything so people know where to listen to. You go into a lot more depth on the testing and the thoughts on coronavirus and all this stuff on your podcast. So that's definitely more thorough, and I would encourage people check that out. This was kind of really an overview, and I am pretty lame when it comes to all of this stuff, so it's hard for me to even formulate questions at times. But I know that when I hear your stuff and other people that are talking about things like you, it's really awakens a lot of curiosity in me because as viruses are just something I've just sort of unconsciously accepted since since I'd first heard about them, whenever that was. And so I think it could be potentially the next new paradigm of sort of discovery. The more people start to open up to it and start to think about it. Is there anything else besides your podcast that you'd want to direct people to? And do you have any sort of like last words on COVID-19, like to help with either things that people should look at or think about if they're still kind of on the fence about what's going on here?
Well, I hope that people keep an open mind and think rationally and not get carried away by what they'd like to think. Like, I think it's always really important to talk to yourself and say, okay, this is what I would like to imagine happens, but I can't let that influence my analysis of the data. I need to analyze the data in as impartial a way as I can. And if that brings me to a conclusion I don't like, well, then maybe some of my fundamental assumptions were wrong. As far as finding more information, I have a website, theinfectiousmyth.com, and a lot of my recent podcasts are there, as well as a variety of papers, some by me and some by some friends of mine who've written articles that I think are interesting on this. So I would encourage people to go there and read the stuff. And I encourage people to read the establishment stuff as well. Read it critically. Read the reports you get from Andrew Cuomo or your local state or provincial government. But look at them a bit more critically about what they're actually telling you and what data is maybe missing that's necessary to fully interpret them.
Yeah.
So,
and you said you're digging into the antibody stuff right now, and are you going to be putting out a podcast covering that stuff, or?
I may very well do that. I will definitely write up a little paper. I've already started writing something up. It's going to be a few days to do this, but I have found a number of papers where they did widespread surveys using the antibody tests, and I have some comments about many of them are not very high quality papers. Some of them are just like press releases from a
Mhm.
from a county government or something, and the people obviously have no idea about. I mean, often they don't even include actual real numbers. They'll say about 500 people tested positive. It's like, well, there must have actually been a number, like 497 or something. Like, why can you not put the number in your press release?
Yeah. Do you have any other coronavirus interviews or things coming up on your podcast or?
Well, I just interviewed Andrew Kaufman. That was this
Oh,
week's
cool.
interview. I have an interview with Stephen Buston, the RT-PCR expert,
Hmm.
with David Rasnick, who's a PhD chemist and drug developer, um, and Kevin Corbett, who's an RN and PhD from England.
Mm,
So yeah, I've had a, a few. Um,
great.
I'm trying to contact somebody who did an interesting analysis of the Gates Foundation, which is a little
Mm,
bit more political than anything else. But a lot of people are talking about the New World Order and all this kind of stuff, and they sort of mix Bill Gates with worldwide conspiracies, and often it ends up they drag in like Jewish conspiracies and stuff like that.
sure. Yeah.
And I'm not going down that road. Um, but this woman had a really dry analysis of what the foundation is really doing and how dangerous it is. And so I thought, okay, well, let's just look at the facts about what this foundation is really doing, where his money's going, where it's coming from, what the relationship is with Microsoft. All of those things are fascinating. So I
Mm,
hope she'll respond to me and we can do an interview on that, because I really think, Bill Gates might not have, I don't believe this was a conspiracy by Bill Gates, but he's sure taking advantage of it.
yeah. 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 I'll definitely keep my eyes peeled for that one because that sounds very interesting. It's unfortunate that some of the, New World Order kind of conspiracy stuff gets wrapped up and kind of tends to shy people away and delegitimize people's, digging into real stuff that's there.
Because there are essential conspiracies where, I mean, to the U.S. government, if you and I have a conversation about a drug deal, that's a conspiracy. It doesn't take very much. Like a
Yeah.
conspiracy doesn't take 500 people.
Yeah.
Um, so I mean, there are people at drug companies behind the scenes saying, we could really take advantage of this to get our drug approved. And I think Gilead's doing that with remdesivir. And
Hmm.
what corners do we have to cut to make sure that we make it to approval before this whole thing is over? Because if the coronavirus epidemic wanes and disappears, our opportunity to get our drug approved is gone, right? So I'm sure there are, what you might call conspiracies about these things. I mean, it's based on narrow monetary concerns,
Yeah.
right? We want to make money for our company. Um, but they're not public, because if people knew everything that a company was saying about their drug, um, for example, Gilead with remdesivir, they changed one of their clinical trials to remove death as an outcome, as a primary outcome. So they changed it for number of days in the hospital, but they only record number of days in the hospital for people who actually leave. If you die, that's not considered.
Mm.
So if there's a higher death rate in their trial, that's not considered a primary outcome. So Gilead can publish press releases saying, according to our primary outcome, remdesivir was a grand success. And then, if you dig into the actual publication, you will find, 13% died on remdesivir and 8% on the placebo. And you go, well, that doesn't sound so great.
No.
But they've achieved their goal because they got press releases to pump up their drug. So there's a lot
Yeah.
of stuff which people need to do, because these drug companies are incredibly unethical.
Yeah, it would almost be nice if there was a different word because calling that a conspiracy just kind of taints it. You know what I mean? But it's just
Yeah,
sort
yeah.
of
It's
it's
not
public
really.
it's public relations, it's having that private thing going on inside the, in the inner workings and then putting a public image that is sparkly and clean.
Yeah, it's not really, I don't want to use the word conspiracy for that. This is just business
Yeah.
for people who've lost their ethical mooring,
Mm.
right? Like
Yep.
and if you're going to be very successful at a drug company, you're successful because you're getting drugs approved or you're selling lots of drugs. You're not successful because you're a moral person.
Yeah.
That's not the route to success.
Yeah. Yeah. It doesn't do the company much good to have a change of heart and decide that, oh, maybe we shouldn't put this drug out that we spent, millions of
Yeah,
dollars.
we're just at the point where we can get it approved, and you say, but we cheated, and then everybody says, well, the FDA didn't notice,
Yeah. Yeah.
so we're not going to tell them.
Oh, well, thanks again, David. I really appreciate it, and I'm definitely gonna yeah, encourage people to check out your website and your podcast and stay tuned to
It
your
was a
stuff.
great pleasure, Jacob. Thank you.
Yeah. Yep.